Hello, and thank you for joining us for this episode of OncLive Peer Exchange. This video series will feature expert panel discussions of current and emerging therapies in the treatment of hematologic malignancies. I'm Dr. Myron Czuczman. I'm Professor of Oncology, Chief of the Lymphoma Myeloma Service in the Department of Medicine and Head of the Lymphoma Translational Research Lab at Roswell Park Cancer Institute. I am also Professor of Medicine at SUNY State University in New York at Buffalo, and today I'm joined by a distinguished panel of physicians and researchers who will offer their clinical insights. Stephen Horowitz is an associate attending at Memorial Sloan Kettering Cancer Center in New York. Lauren Pinter-Brown is clinical professor of medicine at the Geffen School of Medicine at UCLA Los Angeles, California. And Andrei Shustov is an associate professor at the University of Washington in Seattle. And Anas Yunus is chief of the Lymphoma Service at Memorial Sloan Kettering Cancer Center. Thank you all for being here with me today. So let's get started. To start with, we're going to discuss recent advances in antibody-drug conjugates. So antibody-drug conjugates are monoclonal antibodies that are linked to cytotoxic molecules. As we know, effector cytotoxic molecules that are linked to antibodies are typically very toxic for use alone as anti-cancer agents. The ADC technology has enabled the delivery of these highly toxic agents to cancer cells with less damage to normal tissue. Recent advances in linkage technology, along with improvements in selection of these cytotoxins and the use of smaller conjugates with partial rather than complete antibodies, has markedly improved the field of antibody-drug conjugate therapy. One of these, and the one that has the most actual clinical experience we have with, is a CD30-directed antibody-drug conjugate, brentuximab vedotin. This agent was approved by the FDA in mid-2011 for treatment of patients with Hodgkin lymphoma after failure of autologous stem cell transplant or after failure of at least 2 prior multi-agent chemotherapy regimens in patients who are not stem cell transplant candidates. It is also indicated for treatment of patients with systemic anaplastic large cell lymphoma after failure of at least 1 prior multi-agent chemotherapy regimen. So to begin, I'd like to spend a few moments focusing on antibody-drug conjugates from a historical perspective and what the development of the proper type of linker has meant to us. So first I'd like to start with Dr. Yunus and have you give us some of your thoughts and how the actual development of a better linker has improved the field.
Thanks, Myron. So the linker technology is very important for the outcome of the package, which is the ADC, antibody-drug conjugates. You have to have a stable linker that is not cleaved in the serum or the plasma, so you can deliver the payload directly to the tumor cells or the targeted cells without releasing the toxin in the blood, so it will be less toxic. And there's 2 major classes of linkers. One is what's called cleavable and one non-cleavable, and they have advantage and disadvantage. And there's 2 FDA-approved products using either technology. So the cleavable one is, the prototype is Adcetris, which targets CD30. The antibody-drug conjugates bind to the target, internalized, degraded by lysosome, and release a free drug. And the free drug then can be released from dead cells and can then kill by a non-specific effect. So there's bystander effect. Whereas the non-cleavable, when it gets into the cell, it does not cleave a active drug. The drug is still linked to the either antibody or part of the antibody. So when it's released from dead cells, it cannot penetrate a neighboring cell, so it has less bystander effect. And this is the prototype of the HER2 targeted therapy with the M1. So there's advantage and disadvantage for either one of them, and I think you need to look at what disease type you want to. Do you want to have more bystander effect and so forth to choose your linker technology?
Thank you very much. I think what I'll have is a few questions now to the panel. And in particular, since brentuximab vedotin has shown such great success in patients we treat with also their FDA approval, what has been the experience that you've had with brentuximab vedotin in treating heme malignancies? I'll start with Anas, maybe ask you what your experience has been in with Hodgkin lymphoma as well as CD30 positive non-Hodgkin lymphoma.
Right, so as you know, and at the present time, brentuximab vedotin or Adcetris is the only ADC, antibody-drug conjugate, approved by the FDA for the treatment of lymphoid malignancies. It's the first approval came in Hodgkin lymphoma and anaplastic large lymphoma in 2 parallel pivotal trials, showing a very high response rate as single agent in relapse setting. In Hodgkin lymphoma, mainly 100% post-autologous transplant, in anaplastic, both before and after autologous transplant. And that was by itself was sufficient based on unmet medical need to get FDA approval. Now the question is, how do you combine it in frontline regimens, pre-transplant, post-transplant?
What about CD30 positive B-cell lymphomas?
So then, so although the initial trials focus on CD30 positive Hodgkin and anaplastic large lymphomas, because these are the prototype of CD30 expressing cells, but then CD30 is expressed to a less degree in other types of non-Hodgkin lymphomas, and even in some solid tumors like germ cell tumors and so forth. So then there was a trials trying to see what's the efficacy of brentuximab vedotin in these subtypes. So for non-Hodgkin lymphomas, and then I will mention only the B-cell lymphomas, and my colleagues can talk about the other subtypes. So in B-cell lymphomas in relapse setting that express CD30, usually the expression of CD30 is less intense compared to Hodgkin anaplastic, but there is activity. For example, in diffuse large B-cell lymphoma in the relapse setting, there's reported about 40% response rate, which is also durable, about nine months or so median duration of response. So there is activity in other types of CD30 positive lymphomas.